Good afternoon, ladies and gentlemen. We might make a start if that's all right. In the first instance, I'd like to welcome everyone who's come and made time today to enjoy hearing about these two very exciting companies. My name's Michael Woods, and I lead a small advisory firm, Newburyport Partners, and am an executive director of several life science companies, one in the AI and one in the ophthalmic therapeutic device space. And I should note that the session's been recorded, and coming up are two short presentations, both of which are from companies that I've followed for quite a long time and are quite passionate about. So I was very pleased when Rudy invited me to present these two, or introduce these two companies. So there'll be a short presentation and then a time for Q&A, and after that we'll move on to the second company. So the first company I'd like to introduce is called Syntara, previously known as Pharmaxis. Pharmaxis has sold their mannitol respiratory business, meaning that now they're primarily focused on blood-related cancers, and I'll also note scar treatment tissues, or treatment of scar tissue. Global leader in lysyl oxidase enzymes, 3, and I think Gary mentioned now 4 phase 2 trials underway in bone marrow cancer, scar prevention and Parkinson's diseases. They've just completed a $10 million capital raise and on track to deliver significant results in the first half of 2025 with the existing cash runway. So I'd like to introduce Gary Phillips. Joined the company more than 2 decades ago as a chief commercial director, has acted as COO and then CEO from 2013. Prior to Syntara, was at Novartis as group head and CEO of its pharmaceutical division, and has a successful career launching oncology and ophthalmology products. Over 30 years of operational management experience in pharmaceutical and healthcare industry in Europe, Australia and Asia. So I'd like to welcome Gary to the stage.
Good afternoon. So Rudy tells me I've got 15 minutes to talk to you. So, and as you just heard from Michael, I've got 4 phase 2 clinical studies that are due to report results in the next 18 months. So it's very difficult to get through that amount of content in 15 minutes. So I thought what I'd do, I guess you're all here because you're trying to assess opportunities and whether investments are right for you and the right stage. So I'll try and help you with what I think is important at this particular stage of the company and what we're doing, so you can make a judgment on where we're at. So Syntara is the love child, the next company that came on born out of Pharmaxis. So as like I said, I've been with Pharmaxis for 20 years, and for a long time we pursued getting respiratory products, manufacturing them in Sydney and then selling them worldwide. We came to a point where we felt we could no longer support the mannitol business. We didn't think it was going to generate sufficient cash to make it worthwhile holding on to it. And from a shareholder perspective, it was much better course. So what I'm going to talk to you about is the focus that we have now. So Syntara is a company that's focused in clinical drug development. We've got a pipeline. We're experts in lysyl oxidase chemistry, which is an enzyme involved in cross-linking of collagen and therefore fibrosis or scarring, which affects all of our organs in our body, including the bone marrow, liver, lung, kidney, heart, and we've got some anti-inflammatory drugs as well. The selling of the mannitol business has resulted in us reducing our expenses, our cash expenses, on a year-by-year basis by $14 million. So there's a huge weight that's come off of the company. First before the capital raise, where we raised $10 million, and as Michael suggested, that money takes us through to the middle of 2025. And by the middle of 2025, we have not 1, but 4 phase 2 trials that can deliver results. Now, as you know, looking at healthcare companies, phase 2 trial results is where we see not only the safety of the drug, but also whether the drug works or not. They're sometimes small populations, but it's the first indication that we've got a drug that will make it all the way to the market. It's the first real value generating point in a drug's life cycle. So to have 4 of these coming through is almost unprecedented. Those 4 trials are 2 of them in hematological malignancies, one of them in myelofibrosis, the other one in myelodysplastic syndrome. So these are cancers of the bone marrow or blood cancers that cause cytopenia. So looks like leukemia, so reduced red cells, white cells, and platelets. That's 2. The other 2 are, one of them's in skin scarring, where we're working with Professor Fiona Wood in Perth, doing studies in patients with that have had serious burn injuries. They're being skin grafts, and then we're looking at reducing the scarring that comes after the skin graft. And the last one, which is supported by Parkinson's UK, is in a sleep disorder where nearly all of the patients will progress to Parkinson's disease. So it's one of the it's the disease, the predominant disease that leads to Parkinson's, and the challenge has always been to identify, not to treat Parkinson's patients after they get the disease, because by that time they've lost more than half of their dopaminergic neurons. Is to get them earlier on and stop them progressing. So we're treating a sleep disorder, and if we can do that, then we have a commercial drug. But if we had a commercial drug that treated a sleep disorder which also prevented progression to Parkinson's, it would be a double hit. So those are the four studies. I just wanted to spend a bit of time on, because when you're investing, who are you investing with? Who's invested in the company recently? We did a 10 million raise that we closed in December. Our register, we've got a market cap of, I think, about 27 million, 28, when I looked at it before I came in here. So we're a nano-cap company. A nano-cap company with 4 drugs, phase 2 clinical studies, with a register which is 60% institutional. The 2 top ones, DNA Income is a investor that invests in generally. They're based in the UK. They've been following the company for a long time. They've been with us for more than 10 years. They went from 12% to 20% during the last capital raise. The other one is Platinum. Bianca Ogden is the fund manager here in Australia. She's a thought leader in Australian biotech. She went from 8% to 20% in the last capital raise. BVF Partners is a West Coast US company fund which invests only in healthcare companies. They didn't follow their money in the last round. We're just too small, so that their minimum ticket size now is about 30 million US. But they're there and they're not selling, and they want to be there when we get to the next round, which could be in a couple of years' time when we'll have success behind us and where we'll be looking to do much bigger studies. Another 10% behind that, which is again full of specialist healthcare investors. So this is a company with backing from people who invest only in healthcare and tend to look for early opportunities. That's just a slide to say that's the money we took out of the business. We've gone from a company of 65 staff burning around about 5 million a quarter to a company of 25 people, which, including the clinical trial costs, is now burning about 3 million a quarter. So it's a sizable reduction in expenses. We've lost the revenue that we were getting from the mannitol business, but the amount of expenses we've stripped out of the business, and we haven't stopped yet either. So everything about running a small clinical development company is different from running an FDA-approved manufacturing facility. So all of our insurances have gone down. Our systems that we're running are no longer have to be FDA compliant for a GMP facility. So everything is dropping all the time. So I would expect to see more come out as we go on, and we're still looking at the structure and the organization. The board in the company, we've downsized the board. The board is now led by Kathleen Metters. Kathleen was the head of basic research at Merck, Global Merck, for 4 years. She had a budget which is over $2 billion a year and 3,000 staff. We were the first board position Kathleen took up when she gave up. She ran a US NASDAQ listed company before she retired. We were the first board she joined. And even, I took her to actually to see Bianca Ogden at Platinum, and Bianca said to her, why did you choose Pharmaxis or Syntara? What was the thing that triggered you? And she loves the science. She believes in what we're doing. She believes in the science we've got. and has followed it now for several years and was happy to take on the chair. She lives in New York, but ferociously smart lady. I'm really pleased to have her there. Simon Green is ex-CSL. He was the research director of the plasma division at CSL. A stellar career. He's now moved into biotech, and again, we're one of the few board memberships he's got, myself, and then Hashan De Silva, who many of you will be familiar with. He was the analyst at Cars Peak, now setting up his own fund. Again, another thought leader in the Australian biotech space in terms of investments and where to put your money. So, great shareholders, a really knowledgeable board. Investing in this. So, we have 2 streams of drugs, one oral, one topical, that attack lysyl oxidase enzymes, a family of enzymes that are involved in the cross-linking of collagen. So when you injure any part of your body, and that could be through a genetic default, it could be a mechanical injury, a chemical injury, the fibroblasts in your tissues become activated and they release 2 things, collagen and lysyl oxidase. You can think of it like a fiberglass and a hardener. Right? The fiberglass mesh is floppy and soft, but the minute you stick the hardener on it, you form a hard shell. And that's what happens with collagen when you add lysyl oxidase to it. It causes physical cross-links between all the fibers, and that's what gives you scar tissue. Scar tissue is not functional tissue. So wherever it happens, the tissue, whichever it happens to be, loses its function and stops working. So, and as that tissue stiffness grows, it's a feedback loop which then generates more lysyl oxidase and more collagen being released. We have small molecule drugs which completely inhibit lysyl oxidase. They're small molecules, their tissue penetration is excellent. We can measure the level of activity of this enzyme in blood and in tissues, and we can also count the cross-links between the collagen fibers. So we've got assays for all of those things. So in one experiment we can show not only that we have target engagement and that we do inhibit the enzyme, but also that we reduce the number of cross-links, so mechanism of action is also proven. So this mechanism is the same whether we're talking about the bone marrow or whether we're talking about the skin. If we were looking at the fibrosis in the lung for patients with IPF, idiopathic pulmonary fibrosis, or liver fibrosis, or kidney fibrosis, we would find the same thing. Now then, what have we managed to do? First off, the preclinical work has been published in Nature, with 3 Nature publications in the last 2 years. Nature is the highest impact journal on the planet. You don't get your science published in here unless it's at the highest level. We've been collaborating with researchers globally for the last 10 years, and this body of work that's been published is the result of those collaborations. The first indication we're looking at is myelofibrosis. This is a fibrosis of the bone marrow, and that fibrosis leads to low platelet counts, low red cells and white cells. These patients have about 5 years to live from diagnosis, and they're currently treated with something called a JAK inhibitor. A JAK inhibitor does alleviate the symptoms of myelofibrosis, but doesn't change the course of the disease. These patients end up with very large spleens because their bone marrow stops functioning, the spleens take over, and you get lots of abnormal cells being formed, and you get more and more production coming from the spleen, which in turn gets bigger and bigger. So average size of a spleen in healthy patients is less than half a litre. Patients with myelofibrosis often end up with spleens that are larger than 3 litres. JAK inhibitors are good at reducing the spleen size, but they don't get rid of the fibrosis in the bone marrow. They don't restore normal production of red cells, white cells, and platelets, and therefore most of the patients who take these drugs come off of them because they get cytopenia, so they end up with low blood counts of these things. So the idea here is that we can put our drug in, it corrects the fibrosis in the bone marrow, we restore the production of red cells and white cells and platelets, and then these patients can continue on and will have a longer life and less symptoms. We've already tested this. We've used it in a monotherapy sense. This has gone through a phase 2 study approved by the FDA under an IND. We've published this work at the American Society of Hematology last year. More than half the patients that we tested had a 1 grade or more improvement in their fibrosis. So we have proven absolutely that this drug is antifibrotic. So the question is now not, does your drug work, does it reduce fibrosis? The question is, if you reduce fibrosis, do you improve the clinical outcomes for these patients? The answer seems to suggest from this first study, yes. So we saw stable or improving blood counts in platelets and red cells in particular, and we saw improving symptom scores in some patients. This data was enough for us to go back to the FDA and say we'd like to now progress to the next stage, and we've now started a study in combination with the standard of care, a JAK inhibitor. That study is 15 patients. We've already recruited 6 patients. We're due to finish recruitment by the middle of the year. We expect to deliver data at American Society of Hematology meeting in December of this year, and then 12-month data from that study by the middle of 25. Remember, my cash runs out in the middle of 25. So producing data over that period of time is critical, but we're well on the way. We've got 19 clinical trial sites open worldwide, so Australia, Korea, Taiwan, and the US. Those are all recruiting now, 6 patients right now, and I can see the screening patterns in each of the sites, so I'm confident that we'll have all these patients recruited before the middle of the year. So this study's going really well. We've already proven ourselves in the first stage. We're now looking at it. If this study is successful, then we would be back to the FDA at the beginning of next year, looking for guidance on the phase 2B study that would follow. We'd also be talking to just potential strategic partners at that point. Exits for companies with assets in myelofibrosis in the last 2 years with phase 3 data, so one step on from this study, what we'd be going back to do next, have all been above $1.5 billion. There was one 2 weeks ago. Novartis bought a company called Morphosis. They paid $2.7 billion for it. That was a drug with phase 3 data. So there's a huge amount of potential, commercial potential, in this area. So we were really focusing on this and digging into it. The other study that we're doing with the same drug, an oral pan-LOX inhibitor, is myelodysplastic syndrome. It's another hematological cancer. Again, it leads to cytopenias. These are patients who are generally 50 or above. Current first-line treatment is a hypermethylating agent, so azacitidine or decitabine. These again have a long list of side effects they have. The patients here, again, life expectancy of 5 years. As they advance and get high-risk MDS, their life expectancy drops to 1 year. This is exciting because 2 reasons. One is preclinical data from this is absolutely standout. Published in Nature. If you add our drug on top of an azacitidine or hypermethylating agent, you double the response rate in terms of the numbers of patients who get an improvement in their red cells. If you look at it in vitro or in an animal model where you've transplanted the cells from a myelodysplastic syndrome patient. So great preclinical data. Also, fields wide open. So everything in phase 3 at the moment is falling over. And they're falling over because of toxicity. They're causing cytopenias. So whereas myelofibrosis is quite a competitive field, this is wide open. We've got a grant from the MRFF, the Medical Research Futures Fund, which is now funding a study in this, which will start later this year. It will be a study with a dose escalation phase, then a dose expansion phase of 6 months. The first data from this will come middle of 2025, no surprise. So we're hoping that's going to add on. Companies that I'm talking to that are interested in myelofibrosis are also interested in MDS. In fact, they might even be more interested in this than myelofibrosis. So this is really an opportunity to really increase the value of the drug at that stage. So I've told you about 2 indications that we've got. I won't go into the skin scarring. I'm just going to show you the summary of what we have, just to get a feel for it. So this is the list of studies that we're doing. Again, company with a market cap of 30 million, roughly, enterprise value of 17, and cash to complete all of these studies. 4 shots on goal. Myelofibrosis exits north of $1.5 billion in this area, so a lot of commercial interest in it. Myelodysplastic syndrome that will build on that. Scar prevention, I can talk about during Q&A if you like, working with Fiona Wood in Perth. Again, a hugely commercial indication. Just starting a study in that now in patients with burns. And lastly, one in a sleep disorder that's linked to neurodegenerative diseases. So with that, I will leave you. Lots of news flow coming up, and happy to answer any questions you've got.
All right. Any questions? Shall I kick off?
Please do.
So, one question I have reflected on is obviously the fibrotic process is involved in natural processes as well. So what kind of off-target effects and risks? You've passed to phase 1, but still interested in off-target impacts that might materialise potentially over time as well.
Yeah, theoretically, if you inhibited all the LOX enzymes all the time, LOX is involved in the cross-linking of collagen and therefore is involved in connective tissue. So in particular, maybe the tissue that's involved in forming blood vessels, tendons, that kind of thing. So there is a population of patients a population in the US, only happen in the US, that have a diet which is heavy in sweet peas. Sweet peas have a naturally occurring lysyl oxidase in them, and those patients, if you follow them for decades, end up with a greater number of aneurysms than normal. So that's a topic that we follow with the FDA in terms of as a potential safety risk. So the drugs actually stands out at the moment is there are no serious adverse events in any of the clinical studies we've done. We've now been following patients for 12 months, but we've stayed away from using a pan-LOX inhibitor to treat ingrained toenails or things like that. We're sticking with diseases where there's a really high unmet medical need in oncology, where patients have got a life expectancy which you can measure in 1 year, 2 years, that kind of time frame, and all the tests we've done suggest that the only risk to this was if you use this drug in patients who are still growing, still laying down connective tissue. If you only treat adults, then it's a hypothetical risk.
With the Parkinson disease phase 2 testing, is it just conducted in the UK? And that's it?
We've got a site in Australia, in Sydney, and one in Oxford. So that it's a placebo-controlled study, 40 patients. They've all got iRBD, which is a serious sleep disorder where they act out their dreams very violently. I think, as I said, then 90% of these patients go on to develop Parkinson's. And the only thing that we've got to treat these patients at the moment is melatonin, or you try and give them some kind of sedative. So we took this to neurologists, and they thought that it had real potential in addressing this group of patients for two aspects. One is it could actually be a treatment for these patients with this sleep disorder. There are a lot of them. About a third of Parkinson's patients worldwide have iRBD before they get there. So that's still a big body of patients. So there's a commercial opportunity just in giving a treatment for those patients. In this study, we're following them for 3 months, and we're giving them sleep questionnaires to look at their sleep quality before and after. We're also scanning them with a PET scanner and looking at activation of a microglia gland in the brain, which will demonstrate whether the drug is an anti-neuroinflammation drug. If that's the case, which we think it will be, it is in all the animal models, then you could run a study which looks at people with iRBD and follow them to see how quickly they progress to Parkinson's. That study can be done in about 2 years. So the clinicians we're working with can enrich the population and look at people who are fast progressors to try and do it in a reasonable time. So we're not a neurology company. If we get a positive data out of this study, we'll probably be selling it to a CNS company quite quickly, I would imagine. But it's exciting to the point that Parkinson's UK, which is a patient association, gave us $5 million to do the study. So that's the for me, it was like, okay, I don't really know what I'm looking at here, but they went into it in great detail. We had Michael J. Fox Foundation, Parkinson's UK, Shake It Up Australia, all found they tend to work together and make sure they don't overlap. And Parkinson's UK picked up the bill for this one. So.
And no patients from Melbourne at all, just Sydney?
No, they're all patients from Sydney at this point. Yeah, it's one neurologist, and he's using his pool of patients. But anything's possible.
Yeah, hopefully good news. Thank you.
Any other questions?
You appear to have a lot of trials on the go. Which trials are you most excited by?
The myelofibrosis study. I think that's the most advanced. Very clear endpoints that we've got. We're excited by the data we've already seen. The next study is really the proof of the pudding. If the next one is successful and we see good data after 6 months therapy in December, we'll be really on the road then. I mean, commercially it's really valuable, and for patients it would be a huge boon. So, yeah, that's the one. Yeah, if you look at this lot, most of our money is going here. So I think this study is costing us it's probably $5 million before it finishes out of the money we've got. The myelodystrophy, that's coming from an MRFF grant. It costs us $700,000 over 3 years to do that study, so it's not much. The scar prevention stuff is being done with Fiona Wood. We're supporting her, but again, it's an investigator-initiated study, so it doesn't cost us that much. And iRBD is Parkinson's UK. So shareholders have got a chance to profit from 4 studies, which is because we've managed to get non-dilutive funding from other sources to get this range going. Otherwise, we couldn't do it.
All right, I think we might wrap up with you, Gary, and later I think FB Rice will put on some drinks, and if anyone has further questions, they can approach Gary and follow up directly.
Okay.
And move on. Thank you very much, Gary. Right, the second company to come up is Carina Biotech. It's also a company that I was introduced to probably 6 years ago. A really exciting company. I hope Deborah has in the presentation the animation of the activity. It sold me, and if you haven't seen it, I'd recommend you find a way to watch it. So it's an immunotherapy company developing a CAR T therapy for solid cancers, which is more than 90% of all cancers. CAR T, I'm sure most of you are aware, is a revolutionary therapeutic approach using the patient's own immune system and immune cells to fight cancer. They have a phase 1/2A trial of LGR5 targeted therapy for metastatic colorectal cancer enrolling right now, and a world-leading proprietary manufacturing process which reduces the manufacturing time and increases the efficiency, which has always been one of the challenges in the cost of CAR T cell therapy. So I'd like to introduce Deborah Rathjen, the CEO and Managing Director, highly experienced and respected local and international biotech and pharmaceutical executive, having led companies in the US, Europe and Australia. She's achieved success in $800 million worth of transactions and partnerships with major multinational companies such as GSE, Merck, J&J, Abbott, Nycomed, and is the chair of the US headquartered Bioasis Technology and Doherty Clinical Trials.
Thanks, Michael. Unfortunately, I don't have the video of our CAR T cells killing cancer cells, but if you go to our website on the homepage, you'll see a really interesting video there. So Carina is a homegrown company. It's spun out of the Cooperative Research Centre for Cell Therapy Manufacturing. This was a cooperative research centre that brought together all of the researchers across Australia interested in gene and cell therapy, and as the name indicates, was very much focused on manufacturing of these innovative therapies. So really one of the first major initiatives in Australia to get into the field. And that's why we've been able to reduce our manufacturing time. Doesn't really show up on the screen, I don't think, but to 5-day manufacturing time when others are taking 15 to 28 days to manufacture their cells. This obviously helps patients get cells to them quicker, but also reduces the costs. And of course the phenotype of the cells is a much more active phenotype, so really well suited, we believe, to fighting solid tumours, and that's really our focus. Our lead program is a target that has been researched now for some 20 or so years. It has been the subject of other clinical trials through monoclonal antibodies, and there is a company in the Northern Hemisphere that is conducting a trial with a bivalent antibody to LGR5, also targeting another molecule. But really it's an area that's been very well developed. The father of the area was actually Hans Clevers, who a couple of years ago joined Roche as their global head of early stage R&D. So a very well-known target with a lot of biology behind it, and of course we're bringing all of the killing power of the CAR T cell with the very fine specificity of targeting to this stem cell antigen. As Michael's indicated, we're in a phase 1/2A clinical trial. Very early days for us. We're recruiting in Adelaide, in Melbourne and in Sydney, as I said, it's still very early days for us. We have 3 abstracts at the upcoming AACR, and we'll be releasing some information about the trial there. The first indication is in metastatic colorectal cancer, 3rd line. There's very significant need here, and the patients, unfortunately, are very, very common. 3rd line therapy is very poorly active, only a 6% response rate. Other indications include gastric and ovarian cancers, as well as an autoimmune indication, rheumatoid arthritis. So we're privately held and funded through several family offices, and also funded through the 10 Mile Ventures Group, which is part of the Tatarang investment arm of the Forrest family, as well as their Minderoo Impact Fund, in addition to, as I said, several other family offices. So this is CAR T cell therapy. Essentially what happens is that we take the cells from the patient, we engineer them so that they can recognise the patient's own cancer cells. In our case, we're engineering them so that they can recognise LGR5 on their cancer cells. We don't do very much by way of expansion because we get so many cells that we've got multiples of treatments available from a single manufacturing run for each patient. And the secret to CAR T cell therapy in haematological malignancies is it's a single treatment and has the potential to be curative. So that's what we're aiming for with CAR T cell therapy in solid tumours. You can see the revenue forecasts here in terms of the CAR T market and also the estimated percent share of the solid tumours. As Michael's indicated, the solid tumour market is actually over 90% of all tumour types, so there's a big advantage to targeting. Currently there are 6 FDA approved CAR T cell therapies, all for blood cancers, and of course there's been a lot of M&A activity historically in the area, as you can see here, but in the last 6 months of last year there were half a dozen deals done in the CAR T space, with companies like Graycell and Legend Biotech being acquired for several billions of dollars each. So still a lot of activity in the market, and one of the most successful IPOs last month in the US, Kyverna, a CAR T company tackling blood cancer, multiple myeloma, and also an autoimmune disease, systemic lupus erythematosus, SLE, IPO'd at above their price range at $22 a share, and they raised an additional $120 million in their IPO, so from their target. So the IPO market is waking up in the US, and they're waking up to CAR T cell therapy. And by the way, they were early stage, so still phase 1. This is our pipeline. I'm only going to talk about our LGR5 program and why we're so excited about that. But you can see that we have other possible shots on goal with these other programs, including GPC1 and ADAM10, which share some features of the LGR5 target that we're addressing in our initial trials in colorectal cancer. And in that, they're also got features tackling cancer stem cells. We have a collaboration with Adalta on iRBD-enabled CAR-Ts, and in Carina's case, we're interested in being able to make bispecific CAR-Ts. So this is why tackle LGR5. LGR5 is a very important driver of tumour initiation in certain solid cancers, including colorectal cancer, ovarian and gastric cancer. It's really important, it's key to the progression and spread of colorectal cancer beyond the colon and into into the liver and the lungs, for example. And research work has shown that if you can deplete LGR5 positive cells, then you can stop these cancers from forming metastases. But they're also very difficult to cure with chemotherapy and with radiation therapy. Perversely, the chemotherapy that's used in first and second line in colorectal cancer actually stimulates the cells that express LGR5, and I'll show you that this is a very common situation. So colorectal cancer on the rise, particularly in a young population. People as young as 20 are being diagnosed with advanced colorectal cancer. The Australian government recently introduced free screening, dropped the age to 50. In the US, they've now dropped the screening age to 45 in recognition that a younger and younger population is being diagnosed, as I said, at an advanced stage. So there's no CAR T therapy yet available for colorectal cancer, and that's our mission with the LGR5 program. Overall, the therapeutics market for colorectal cancer is approximately 10 billion US per annum, and it's growing at about 5% per annum. So this is what a colorectal cancer that's spread to the liver looks like, and the brown indicates that this tumour is very, very full of LGR5, and you can see the magnification here. Very low magnification to see the staining, very high magnification to see any staining in the colonic crypts with LGR5. Not much of it about. So we've investigated this further at Carina, and we've now got a patient population of over 500 people that have been evaluated looking at their LGR5 levels when they've got primary colorectal cancer, and you can see that pretty much everybody has LGR5, low levels of LGR5 in many of them, but about 60% of patients have medium to high level of expression of LGR5. This is a very important slide because it shows you that what we're doing here is actually clearing the cancers totally. And this result is persistent, so a single inoculation of CAR T cells into these animals presents with a very durable response out beyond 100 days. And if we try to inoculate these animals with new cancer cells at around 42 days after their initial treatment, the tumours do not become established. So that really speaks to the curative potential of LGR5 CAR T cells, but also the fact that they've got this memory, this immunological memory. We know that these CAR T cells persist in the spleens and other lymphoid tissues of these animals for prolonged periods of time. I've actually talked to this data. So here's the flat line showing complete clearance, both as a combined group and as the individuals. So you can see there were no breakouts at all, and you can see that translates to complete survival of the LGR5 CAR T treated group. This is the re-challenge that I've just talked to. So you can see this is where they've been given the initial challenge here. Then they've been given another challenge with tumour cells here, and this is the result. So no tumours appearing. So we get durability of response, we get immunological memory. That means that if the tumours come back at all, then there are CAR T cells that can be activated to fight the cancer. This is an outline of our clinical trial. It is a phase 1/2 clinical trial. We are at the early stages, so still at dose cohort 1. I'll share with you just some early observations about that. And you can see that we've got our clinical trial on clinicaltrials.gov. We're only enrolling patients that express high levels of LGR5 into our trial, so only those patients that are able to respond to an LGR5 depleting agent are being enrolled. And we had to develop an immunohistochemistry assay for this, very tricky. We looked at over 30 antibodies to LGR5 to find ones that could be used in immunohistochemistry. We're paralleling that by looking at LGR5 at the RNA level as well, but the immunohistochemistry is the important one. And this, of course, could lead to a diagnostic therapeutic combination in the future. So, as I said, we're in the phase 1 component, where of course we're looking at safety and tolerability. We're focused on identifying the recommended phase 2 dose level. Then we can open up the trial. We expect once we get to phase 2 that it will enrol within a 6-month period, because these patients are so frequent. Here's patient 1. And I'll talk a little bit about her. So you can see immediately that you don't need to be a pathologist to identify in her liver biopsies where the LGR5 positive cells are. You can basically hold the slide up to the light and you'll see it, but nonetheless there's an independent pathologist that reads all of this. Okay, 1 minute. Okay, so she's doing well. She's 3 months on study and doing well. And of course, we've got a whole pipeline of patients now out through to dose cohort 2. This is our manufacturing process. I want to tell you 5 days, really great cells, high transduction, that's the secret. And then the other secret is getting this profile of cells. And then we've got other things in our toolkit that help the CAR T cells get where they need to be and do their job, that are proprietary to Carina. So that's it. Sorry if I've gone over time, Rudy. Yeah, great.
All right. Any questions for Deborah from the audience?
Any plans for going in public? Or?
Yes, Rudy said you'll get asked that question. So, you know, we're always keeping an eye on it, and have been, you know, quite pleased to see Carina's positive IPO last month in the US. We're watching another couple of companies in the US and see how they go. Had discussions with the ASX towards the end of last year about going public. Not so encouraging was the ASX, so we'll have to keep watching there and waiting to see what feedback they give us. But well, they're really wanting companies that are at a little bit more advanced stage than we are. Yes, yes, that's right. Yes, but as I said, Carina got away in the US in phase 1 in multiple myeloma and phase 1 in SLE. So, you know, and also, you know, by the middle of next year we'll be in phase 2, maybe sooner, and again we will have maybe passed that hurdle. So yes, we are planning to become a public company at some point in some jurisdiction, and we'll look to see where that works best.
There's another question over here.
The iRBD product,
Yep.
is that with 1AD, is it?
That's with Adalta, yes. So we're collaborating with them. They, under the agreement, they have their targets of interest and we have our targets of interest, but we share technologies.
Any other questions?
Where do you conduct your trials?
That's a really good question, because we did go to the US to file an IND, and we so our IND was cleared by the FDA, but we are running the trial here in Australia. So there's a centre at Peter Mac here, and centre in Adelaide at Royal Adelaide Hospital, and in Sydney at the Kinghorn Cancer Centre. And our cells, the CAR T cells for patients, are manufactured here in Melbourne as well. So it's very much an Australian effort.
There's a lot of attention on some of these cancers, particularly the colorectal cancer. and there's several trials recruiting around. Are you finding there a competition for patients to participate, or is that an exclusion criteria for you guys if somebody's been on a trial of something else and it hasn't responded?
Yeah, there are exclusion criteria, and that so that we look for more than just are you LGR5 positive. They do have to be treated with certain chemotherapies, which comprise the first and second line therapy. So we're going third line, but there's absolutely no competition for the patients. And in fact, my big concern is actually more about manufacturing, because when we get to phase 2, the number of patients that are available will potentially outstrip the manufacturing slots that are available. So that's my big concern that we're working on at the moment.
All right. Any other questions?
These companies that were picked up by Big Pharma, the
Yep.
2, 9 billion, 11 billion.
Yep.
What do they have, or what did they have at the time that they were acquired
Yep.
that you don't currently have?
Well, they had some data in the blood cancers. So those two companies discovered and developed the first 2 CAR T cell therapies that were ever developed. And it was the first time actually that CAR T therapy had been shown to be curative. They were treating children with blood cancer, and those children are alive today, some 10, 11 years later, leading happy and healthy lives. So it's a matter of having the data in the clinic to show that yes, your therapy is doing what it's supposed to do in terms of safety and efficacy.
And how long potentially would it be till you would have that data?
Well, that's really interesting, because that's a very interesting question, because what we're finding at the moment is that Big Pharma wants to move into solid tumours. They want novel targets. So Carina, solid tumours tick, novel targets tick, and to varying degrees they want to see data. Now, some are more interested in being there right at the beginning than others, but typically they want to see that it's safe and that you've got a path forward. So that's getting to your recommended phase 2 dose level. So, the next 12 to 18 months should be pretty interesting for us. Yeah.
Are those deals on whole portfolios or individual cancer indications?
Well, the ones over the second half of last year were company acquisitions.
Yep.
Yeah. But as I said, still at the early stages, so still phase 1. Yeah.
Any other questions? All right, we might thank Deborah for her time, and thank all of you audience for attending. The session today was recorded and will be on Monsoon's YouTube channel. So if anyone wants to go back and review the discussion or the slides, I'm sure they'll be visible on that. And so we also should thank Ian for hosting us all and putting on some refreshments later at FB Rice. And I welcome everyone to stay and have some refreshments and follow up with Deborah or Phil later if they want. Gary Phillips, sorry.